Okay, so this EdPuzzle is going to focus on the process of endosymbiosis, which describes how we think all eukaryotic cells ended up with mitochondria inside them, and how the land plants and their relatives ended up with chloroplasts inside their cells. We're going to walk through the process step by step, so I encourage folks to take good notes because you might be asked to describe the steps involved in this process on a free response question on the exam, possibly. So make sure you know what's happening at each step, which parties are involved, which type of cells are involved, and the outcome of every step in terms of how it may benefit both of the symbiotes involved in the process. We're also going to review the evidence that we have that supports this particular theory, and then we're going to go over what a secondary endosymbiosis is and how it's different from primary endosymbiosis and how it affected the evolution of eukaryotes in general.
Okay, so how did we get from a cell that was like an ancestral bacteria or ancestral archaean to the first eukaryotic cell? And so our best educated guess as to how this occurred, right, is you start with your nice prokaryote cell, and possibly during cell division or some other part of this process, you get the plasma membrane to sort of make little pockets or folds in itself, and those folds kind of pinch off and become vesicles and migrate and then merge together to where you have a layer of membrane around the genetic material. There's our nuclear membrane or nuclear envelope. And then you can get other important organelles like the Golgi, endoplasmic reticulum, and all those kinds of things from that. We also know that somewhere in here, right, cytoplasm DNA at some point, right, we went to, if I can get my pen to work, there we go, right, so linear chromosome. From a circular one. It's supposed to be an O. Oh, that's a mess, isn't it? Oh, that was weird. Okay. Okay. Sweet. All right, so you had a change in the structure of our DNA at some point. You had all this folding that produced, again, over a long period of time. Remember, we had, I don't know how many, millions of years. I think it was actually in the billions between our first prokaryotic cells and our first eukaryotic cells. We can go back and look at the cosmic calendar there to get more information. But you have the fusion of those membranes around the nucleus, right? We've got our nuclear envelope here with our endoplasmic reticulum, and of course all of our other organelles. And then something rather remarkable happened. And you have, we have to remember, right? This is a heterotroph, right? So our ancestral prokaryotes, right, where we had lots of variety of lifestyles, right? You had some that were autotrophs, they made their own food, and some that were heterotrophs. And so here we go. I'm going to put it right. Well, this is actually our ancestral heterotrophic eukaryote is actually right here because he's engulfed something. So we have our ancestral. Actually, let me just do this. I'm having too much fun playing this with this pen. Okay. So here's our ancestral heterotrophic eukaryote, right? So he doesn't make his own food. He ingests other organisms or organic matter, right? And then breaks it down. So at one point, right, so engulfed essentially equals ingested, basically ate it by a process called phagocytosis. What's going on here? There we go. This is the same process, right, that amoebas still use. Our white blood cells still use this to deal with bacterial invaders. You wrap it in membrane and bring it inside the cell. And that's essentially what our ancestral eukaryote did. They wrapped it in membrane, whatever it was that it ingested, probably a proteobacteria, wrapped it in cell membrane, and then normally, right, you would merge that with a lysosome or something that's containing digestive enzymes. For whatever reason, either that didn't happen or it didn't get digested properly or something, but basically that proteobacteria wrapped in membrane started living inside the host cell. You ended up with a symbiotic relationship, right? So for whatever reason, right? So proteobacteria, so somehow not, this should be a W, not digested, and now it's living in eukaryote cell. And we have our mitochondrion, right? This is our symbiont that we can't live without anymore, right? It's now obligate. We actually really, really need that guy. We can't survive without it. So this helps explain how all eukaryote cells have mitochondria. Okay, so now that all of our eukaryote cells have mitochondria, so this is where we left off, right? So this is from the previous slide right over here. So some of these guys, though, sort of went on their merry way. We have a divergence point. So right here we have lineages diverged. And so we still have heterotrophic eukaryotes. And then we have photosynthetic eukaryotes, right? But you'll notice that these guys already have the mitochondria, so that we know, right? This diverged after proteobacteria was ingested. Because all eukaryotes have mitochondria, but only some of them have chloroplasts, right? So we think that our, let's see, engulfed photosynthetic bacterium, probably a cyanobacteria, for the reasons I outlined earlier when we were looking at cyanobacteria, right? You see all the folds in the membrane that are pretty much identical to the thylakoids. They use the same pigments for photosynthesis, chlorophyll A and B. And so we just think we have repeated repetition of this previous process, right? So you had phagocytosis, ingestion, didn't break it down, started living inside the host cell. Now you have a symbiotic relationship where both parties win. And so the one branch goes this way. This, of course, is all of our heterotrophic eukaryotes. And then you have another branch going this way that also has the mitochondria, but has now engulfed the photosynthetic bacteria. And so cyanobacteria ingested, but not broken down like before, and you end up with, right, so this is a chloroplast. Same thing, it's living inside, right, this is a symbiont. Living inside the host cell. So this, of course, is our ancestor of plants. So we have an autotrophic photosynthetic eukaryote. Okay, summarizes that pretty well.
What if you could absorb another organism and take on its abilities? Imagine you swallowed a small bird and suddenly gained the ability to fly. Or if you engulfed a cobra and were then able to spit poisonous venom from your teeth. Throughout the history of life, specifically during the evolution of complex eukaryotic cells, things like this happened all the time. One organism absorbed another, and they united to become a new organism with the combined abilities of both. We think that around 2 billion years ago, the only living organisms on Earth were prokaryotes, single-celled organisms lacking membrane-bound organelles. Let's look closely at just 3 of them. One was a big, simple, blob-like cell with the ability to absorb things by wrapping its cell membrane around them. Another was a bacterial cell that converted solar energy into sugar molecules through photosynthesis. A third used oxygen gas to break down materials like sugar and release its energy into a form useful for life activities. The blob cells would occasionally absorb the little photosynthetic bacteria. These bacteria then lived inside the blob and divided like they always had, but their existence became linked. If you stumbled upon this living arrangement, you might just think that the whole thing was one organism, that the green photosynthetic bacteria were just a part of the blob that performed one of its life functions, just like your heart is a part of you that performs the function of pumping your blood. This process of cells living together is called endosymbiosis, one organism living inside another. But the endosymbiosis didn't stop there. What would happen if the other bacteria moved in too? Now the cells of this species started becoming highly complex. They were big and full of intricate structures that we call chloroplasts and mitochondria. These structures worked together to harness sunlight, make sugar, and break down that sugar using the oxygen that right around this time started to appear in the Earth's atmosphere. Organisms absorbing other organisms was one way species adapted to the changing environmental conditions of their surroundings. This little story highlights what biologists call the endosymbiotic theory, the current best explanation of how complex cells evolved. There's a lot of evidence that supports this theory, but let's look at 3 main pieces. First, the chloroplasts and mitochondria in our cells multiply the very same way as those ancient bacteria, which are still around, by the way. In fact, if you destroy these structures in a cell, no new ones will appear. The cell can't make them. They can only make more of themselves. Second piece of evidence. Chloroplasts and mitochondria both contain their own DNA and ribosomes. Their DNA has a circular structure that is strikingly similar to the DNA of the ancient bacteria, and it also contains many similar genes. The ribosomes, or protein assembly machines of chloroplasts and mitochondria, also have the same structure as ribosomes of ancient bacteria, but are different from the ribosomes hanging around the rest of the eukaryotic cell. Lastly, think about the membranes involved in the engulfing process. Chloroplasts and mitochondria both have 2 membranes surrounding them, an inner and outer membrane. Their inner membrane contains some particular lipids and proteins that are not present in the outer membrane. Why is that significant? Because their outer membrane used to belong to the blob cell. When they were engulfed in the endosymbiosis process, they got wrapped up in that membrane and kept their own as their inner one. Surely enough, those same lipids and proteins are found on the membranes of the ancient bacteria. Biologists now use this theory to explain the origin of the vast variety of eukaryotic organisms. Take the green algae that grow on the walls of swimming pools. A larger eukaryotic cell with spinning tail structures, or flagella, at some point absorbed algae like these to form what we now call euglena. Euglena can perform photosynthesis, break down sugar using oxygen, and swim around pond water. And as the theory would predict, the chloroplasts in these euglena have 3 membranes, since they had 2 before being engulfed. The absorbing process of endosymbiotic theory allowed organisms to combine powerful abilities to become better adapted to life on Earth. The results were species capable of much more than when they were separate organisms. And this was an evolutionary leap that led to the microorganisms, plants, and animals we observe on the planet today.
So an important thing to remember is what else was happening in the world at the time that first heterotrophic ancestral eukaryote ingested that proteobacteria, right? So about that time, about the time the first eukaryotes were evolving, right? Remember, we had the oxygen revolution, right? So you went from a relatively anaerobic environment to suddenly, a decent amount, a proportion of the environment is oxygen, right? Which of course is toxic to a lot of anaerobic organisms. You had these mass extinction, mass die-offs, and we believe, again, the proteobacteria, based on the characteristics of the mitochondria that we have, were likely, here we've got a proteobacteria that can not only exist in an aerobic environment, but actually use that oxygen to, through the process of cellular respiration, use that oxygen to extract the energy in the bonds of organic molecules and make energy, essentially. So like we saw in the little video, right? You had the little guy that shakes, oh, I'm making energy, yeah, this was a huge breakthrough, right? If you are that ancestral eukaryote, boy, you hit the jackpot by ingesting this guy. Thank goodness he didn't get broken down, right? Which who knows how, this is only believed to have happened once. So clearly a rare event. And so again, you can not only handle that toxic environment, but actually get something good out of it. And now you've got a little bacteria that's living inside the host eukaryote cell. He's getting protection, he's getting nutrients. So it's really a win-win situation for both of them. And the evolution, of course, continues, and they become more and more dependent on each other. And of course, now we can't live without our mitochondria. So of course, shortly thereafter, we had one lineage diverged, and those are our heterotrophic eukaryotes with mitochondria. And then we have another branch, right, that became the ancestors of plants. So these guys also, of course, have mitochondria, but then we had another ingestion event. This time, the cyanobacteria that got ingested, wrapped in membrane, brought inside the cell, didn't get broken down. And of course, there was a benefit to keeping the cyanobacteria as it was, right? It can do photosynthesis. Now, it provides oxygen. Well, guess what? You don't have to worry about that because you have mitochondria that can use the oxygen to break down the sugar. You have a cyanobacteria, which again we are very confident is the source there of the chloroplast. You have the cyanobacteria, cyanobacteria that are producing sugar, so they're essentially producing even more energy. And it's again a win-win situation for both of them involved, right? Both the chloroplast gets a nice place to live, it gets all the nutrients that it needs, right? All the organic phosphate and nitrogen that it needs, and the rest of the cell gets energy out of this process. And suddenly you have a lot of energy, right? You're getting energy from that. You can get energy from mitochondria. And it's like, wow, I don't have to ingest stuff anymore. I am now a photosynthetic eukaryote. And now I can really start to get more complicated too. Same went for our heterotrophic eukaryote lineage. It just took them a little bit longer to get there.
Okay, so what evidence do we have to support this theory? This theory that was sort of widely mocked at the time that it was proposed by Dr. Juliana Margulies, and is now, of course, widely accepted as the most likely explanation, scientific explanation, for how eukaryotic cells came to have mitochondria and how the ancestors of land plants and plants ended up having chloroplasts inside them. I posted an additional video to Canvas called the Endosymbiotic Theory. Not only does it have nice catchy music, but it gives a little bit more background on the theory and the evidence to support it, so it's a nice additional recap. So evidence to support this. Some of the biggest pieces of evidence, of course, are the fact that both mitochondria and chloroplasts are wrapped in two membranes. So the inner membrane is homologous with a prokaryotic membrane. It has prokaryote proteins embedded within it. And the outer one is a eukaryote membrane, meaning it has eukaryotic proteins embedded in it and is thought to be the remnant of the vesicle that was used to wrap that during phagocytosis to wrap the bacteria in and then bring it inside the cell. So essentially we have the inner membrane is the original cell membrane of the bacteria that was ingested, and the outer membrane is the packaging that it was wrapped in during the cell eating process. Mitochondria and chloroplasts also replicate themselves in the same way as prokaryotes. They basically use binary fission. They don't do, because again they have a circular chromosome and not a linear chromosome, so they don't do mitosis. They also divide separately during cell division from the rest of the cell. And of course, I think as I had mentioned, I will mention later, they replicate themselves separately because they have a circular genome, but our cells don't contain the instructions on how to make more mitochondria. So they have to basically copy themselves. Our cells contain instructions on how to make all of our organelles, all of our other cell components, but not our mitochondria. And an additional piece of evidence for this theory is the fact that chloroplasts can engage in horizontal gene transfer with any bacteria, symbiotic bacteria that live in the cells of, for example, nitrogen-fixing plants, right? Have rhizobacteria. Rhizobacteria can exchange genes with the chloroplasts in their host plant cells. And of course, we know that only prokaryotes can really engage in horizontal gene transfer. I think there have been a couple of cases documented of eukaryotes using horizontal gene transfer, but it's extremely rare, and those may have been some really interesting special circumstances. But we know the horizontal gene transfer is very commonplace amongst prokaryote cells, right? So bacteria and archaeans can exchange genes with each other. Different species of bacteria or different species of archaeans can exchange genes with each other. So the fact that rhizobacteria can exchange genes with chloroplasts really does suggest that they are both prokaryotes, or at least one of them was a prokaryote at some point, and still has that capability of exchanging genetic information. We actually take advantage of this in some genetic modification. For example, trying to give certain plant strains resistance to, say, frost or some diseases. We can put genes into a bacteria and then essentially expose or inject that bacteria into the plant cells, and the chloroplasts will exchange some of the genes and hopefully exchange some of the genes that are for frost resistance or disease resistance with these new introduced bacteria.
And of course, they can replicate themselves because they have their own DNA, their own instructions. What's interesting is, and people have done this, they've experimentally removed both mitochondria and chloroplasts from plant cells, as well as mitochondria from other eukaryote cells. And the cell is not able to manufacture those components again. Once they're gone, which of course very much supports this idea that these guys used to be free living organisms, and now they're living inside. It's now an obligate situation, right? They can't live. The mitochondria can't live outside our cells either. But our cells can't function. They can't make mitochondria. So not only do mitochondria contain their own DNA and their own ribosomes, but the genome is a circular chromosome, which is like prokaryotes, right? We know that was one of those we talked about last time, right? One of those changes that occurred in very early on in eukaryote cell evolution is going from a circular genome to a linear genome. Well, mitochondria and chloroplasts have a circular genome, just like modern prokaryotes. And their ribosomes are much more similar, almost identical, to the ribosomes of prokaryotes. They are very different than eukaryote ribosomes, which is interesting, down to their sensitivity to antibiotics, right? So if you are a person that's been on antibiotics, maybe if you got a resistant infection and you've been on antibiotics for months and months and months, those drugs have probably starting to leach. Again, they've got to go through, your one cell membrane. They've got to go through the outer mitochondrial membrane. They've got to go through the inner mitochondrial membrane. But they get inside your mitochondria, they can start affecting your mitochondrial DNA. They can start affecting, not the DNA, excuse me, the ribosomes and the function of your mitochondrial membranes. And so folks, again, who've been on antibiotics for a really long time tend to be very, very tired as a result of the mitochondria damage. A heterotrophic eukaryote cell with mitochondria, right, could survive in aerobic conditions. And of course, not only survive, but have extra energy for growth and development, extra energy to increase complexity. Same goes for our first photosynthetic ancestral eukaryote who had mitochondria and then now chloroplasts. So all that extra energy allowed them to, again, become larger, more complicated, and resulted in, eventually, multicellular organisms in multiple eukaryote lineages. So this was a really big breakthrough. And notice that prokaryotes, right, are still single-celled, unless you count the filaments like cyanobacteria and actinomyces and things like that. But for the most part, right, single-celled organisms. Eukaryotes, right, can be huge. Largest living things on the planet are eukaryotes. So having all that extra energy really allowed for a lot of a huge increase in complexity in eukaryote evolution. Okay, so what the heck is secondary endosymbiosis? If this process of acquiring mitochondria and chloroplasts is primary, then what is secondary? So basically, secondary endosymbiosis, right? Primary is a eukaryote ingesting a prokaryote. Secondary is a eukaryote ingesting another eukaryote. But in this case, specifically, a eukaryote from the supergroup Archaeoplastida, basically ingesting a eukaryotic cell that it has already acquired chloroplasts or plastids. And this helps explain how you can end up have organisms that are not very closely related at all to plants, but that have plastids inside them. So they have either ingested a green algae or a red algae, and then you have the same process, right? You're a heterotrophic eukaryote. Excuse me. You ingest something. Well, the first thing you do is you wrap it in membrane, bring it inside. And the same process again. There was a huge benefit to holding on to that eukaryotic cell and not allowing it to be completely broken down. It did get broken down partially and in different lineages to different degrees. In some cases, there's hardly any of the eukaryote cell left. In some, there's more. But essentially the same situation where the plastids start living inside the heterotrophic eukaryote cell. And now that cell has acquired those characteristics of the algae that it ingested. Okay, so exactly how does this process of secondary endosymbiosis work? And the big part of this graph that I always find terribly confusing, so here we've got cyanobacteria, right, being ingested by a heterotrophic eukaryote, right? This is primary endosymbiosis. And then it's living inside and it says, hey, there's 3 membranes. And we're like, well, we just discussed how there's only 2, right? What do you mean there's 3? Well, my understanding anyway is that proteobacteria was probably a gram-negative bacteria, so it would have that third membrane layer or that second membrane layer on the outside of the cell wall, right? And then you wrap that in membrane, that's 3 membranes. And so they lost one of these, and that's why you have only 2 membranes around our chloroplast here. And then, of course, you had you had eukaryotic cells ingest that green algae and multiple lineages, right? So this has happened multiple times. So here we've got our ancestral algae. I'm just going to put this right here. So our ancestral algae. And then you have a you have a speciation event, you have a divergence, right? And so we get one trajectory that goes to red algae, right? This is just a change in photosynthetic pigment. And these guys are still using chlorophyll A and B. And then you have, this occurs multiple times. In one lineage, we get somebody eating these guys. It goes to this, however the heck you pronounce that, right? And then we have another example where we have the euglenozoans ingested a green algae and wrapped in a membrane. And now it's living inside their cells and they can do photosynthesis. They're also still heterotrophic, so what we call mixotrophs. They can do both. Don't worry about these guys. If I can get, there we go. Actually, that's kind of a mess. Let's try to make that a little bit cleaner if I can. Or not. Anyway, don't worry about those guys. We're also not going to be covering these guys, which is another, so we've had multiple lineages go through secondary endosymbiosis with red algae as well. And then, of course, the other branch is our dinoflagellates, which we are going to talk about next time.